We're really fortunate this afternoon to have two of the luminaries in metabolomics giving talks on their research and applications, Dr. Oliver Fiehn and Dr. Lloyd Sumner. And then after their talks, we're going to have about a 30-minute discussion section, which some of the other speakers will also be able to join from other sessions of the symposium. And really that's for anyone who's participating to ask questions or just to have a general discussion on the future of metabolomics wherever the audience wants to go. So without further ado, I'd like to start by introducing our first speaker of the afternoon. That's Dr. Oliver Fiehn. So Dr. Fiehn obtained his master's, well, his diploma, it's equivalent of master's in the German system, at the Free University of Berlin in Germany, of course, and then received his PhD in 1997 in analytical chemistry at the Technical University of Berlin. He was a group leader at one of the Max Planck Institutes, the Max Planck Institute for Molecular Plant Physiology. That's in Potsdam, a really wonderful institute I visited once several years ago, back when we could still travel. So he was there from 2000 to 2004. And then he moved to UC Davis, where he is a professor in the Genomics Center and also in the Department of Molecular and Cellular Biology. And most importantly for this symposium, he's the director of the West Coast Metabolomics Center. Dr. Fiehn has been really instrumental in developing and standardizing metabolomics databases and libraries, tools that all of us in metabolomics use. So in particular, we heard this morning about MONA, the Mass Bank of North America, which is freely accessible. But that's just one of the databases he's been involved in developing that we all are benefiting from. And in 2014, Dr. Fiehn received the Lifetime Achievement Award from the Metabolomics Society. In addition to all that, he has his own research aiming at understanding metabolism across all the domains of life, including both human and plant and microbial systems. And today he's going to focus on humans. And the title of his talk is How to Interpret Metabolomics Data from Human Cohort Studies. So take it away, Dr. Fiehn.
Thank you. All right. So welcome, everyone. I'm glad to be here with you. It's fine to post questions anytime, and I do this more in a workshop style, so that's not really a research presentation, rather than maybe learning from research that we have done and exemplifying what and why we have done it, a little bit also in the style of tutorial. So I will focus here on human cohort studies, and Professor Lloyd Sumner will then talk about plant studies. So the context for clinical metabolomic results obviously deals with humans. Clinical means these are studies that are on people. If you have mice or rats or dogs, these are not clinical studies. Cells, not clinical studies. Clinical studies deal with humans. Okay, and when we look at metabolomic results from clinical studies that come from human, most of the time, they come from clinicians, of course, and most of the time we get biofluids like plasma, serum. Sometimes we get saliva or urine. Rarely we get CSF fluids from the brain, but usually we get fluids. Sometimes we get tissues, biopsies, specifically in cancer metabolism when there's surgery. So we always have to bear in mind the context of such clinical studies and when we try to interpret things. So this is also true, of course, for other systems, like other multicellular systems, but specifically for humans, where we get plasma. Plasma is a transport system of metabolites, but these metabolites can come from various sources. Some of them may come from export from cells in some tissues. Others may come from the digestive tract, calorie tract, and food composition. And of course, these are influenced by our gut microbes that, convert food compounds, and, the liver, also converting food compounds. And all that is overlaid then by genetics, SNPs, sex differences, methylation status, and so on. So when we have data sets that come from clinical metabolomic results, this is the context. And the very first question we then have to ask ourselves is, what are we trying to do here? Are we trying to map pathways? Because from plasma, that's very difficult, because that is a very indirect way of trying to go to metabolic pathways, genes, not only because the cells have transport systems, lots of transport systems, but also because pathways in cells are happening in organelles. Some of that is, of course, in the cytoplasm, a lot in the mitochondrion, but even the nucleus has pathways, and certain, the lysosomes have pathways, the peroxisomes have pathways, and of course the endoplasmic reticulum has pathways. So just mapping metabolites to pathways is not the answer for clinical metabolomic results. And often that is not even the question. So we really have to interact and engage with the clinicians to find meaningful answers to their questions. And that is meaning that this cannot be done by the clinicians themselves. It can also not be done by the analytical chemists themselves. And it certainly cannot be done by the statisticians themselves. So clinical metabolomic results, even more than other biological studies, make it necessary for teamwork, for collaboration and discussion. So what are the typical questions we ask in human cohort studies? Can be broadly defined into two things: either diagnostics. So where diagnostics, the answer of metabolomic results might be leading or possibly leading to diagnostic decision-making, patient stratification, or used in public health, like for risk factors, diabetes risk factors, for example, that can then lead to policymaking. But by and large, that is where people then say, we just want a biomarker. But you have to be very, very clear if you say that. What type of biomarker? How good must the biomarker be? And what are the underlying other types of biases or disease routes that can be looked at? So a biomarker for healthy versus disease is usually not a clinical question. Usually a clinical question is, which drug should I do? Which therapeutic options do we have? What are the likely outcomes when I have a certain stage in the diagnosis, for example, cancer 1, cancer 2, or stage 3 cancers, right? So these are typical questions in the clinic, not just having a disease versus healthy populations. But often enough also, the idea of clinical studies goes beyond that. So that we try to really not only get biomarkers of some sort, but also we want to improve our understanding, both on the cellular level and the physiological level, potentially being able to give advice to patients, at least add to the mechanistic knowledge in the clinical arena, and possibly utilize that as input into new therapeutic options. And that means really we have two things to do. First of all, statistics. in diagnostics, it's mostly a statistics question. Not exclusively, but really heavily relying on statistics. But when we try to answer mechanistic questions, we have to go beyond that. We have to do first the statistics, but then we have to stick our heads together and really try to interpret, based on the slide what I've shown before, all these interactions of genomes and microbiomes and food and exercise and health disparities, backgrounds, and cellular pathways. It is therefore not as easy as just taking a data set and mapping it. And by the way, I am not talking here about anything that an analytical chemist would do. I here assume that you already have somehow a good data set. Either you generated that in your laboratory, or you received it from a service laboratory, or you received it from a commercial entity. There are certain metabolomic commercial entities that can give you data. So that's my assumption here, that you already have this data. I'm not talking about how to make good data. So once you have good data, you give it to the statisticians. And then you hope the statisticians will tell you what's going on. Well, I can only tell you, do not leave statistics to the statisticians. Engage with them. And the reason is, statistics is a science, a field of science with lots of options. Just like you have many options in biology and many options in analytical chemistry, so do you have many options in statistics. That means statisticians ask questions to data, and we have to guide them what are relevant questions, what are good questions. And it's not just sending data sets out and getting a table back. So if you do this, you will get the following. Assume now you have 1,000 identified metabolites in a typical metabolomic data set. That's today true. And let's assume that you have a bunch of actually true biomarkers and a bunch of actually not true biomarkers, like compounds that are not relevant to the clinical questions you have. And what a typical clinical statistician will do is they will try to have as many true values without any false values. So they will take this threshold here that distinguishes the two populations of possible values or metabolites, and put this threshold as far out to the right-hand side as possible. Not sure if you see my little arrow here. That means necessarily you increase the false negatives. So because these ones that are shaded here in green, we just define them as actually true biomarkers, but they would have an overlap in statistics in p-values. And the reason is, usually clinical statistics comes from diagnostics, not from mechanisms. And diagnostics, because it's life and death situations, cannot or must not have false positives. So that's why, by default, clinical statisticians would put that threshold very much far out to the right. And in epidemiology, they would also have lots of adjustments for sex, body mass index, and many other things. That's why they then use false discovery rate settings to fight the multiple assessment problem. So if you have 1,000 metabolites, just by random bad luck, at a p-value of 0.05, 50 of them will be actually not true, and you just don't know which one. And you can't just say, oh, I just make a higher threshold of p-values. So that's the one thing. So that's why when you just leave it to statisticians, that's what happen. They will take your data, apply a hard FDR correction, usually a Benjamin Hochberg or something else, and you will return very few single compounds, because that's what they do. So, however, these hard-cut false discovery rates therefore necessarily get you a lot of false negatives, right? So what then mechanistic people do is they say, well, we just want to have as many true positives in it. And then let's just take the raw p-value of 0.05. So we switch this threshold over here and, really get the whole suit, the whole population of true positives. But that necessarily means we also get a lot of false positives if we do it this way, right? So that is a conundrum in classic univariate statistics. All right. So 20 years ago, metabolomists said, well, that's why we use multivariate statistics. We use the whole panels of metabolites, distinguish them, and then we don't need FDR corrections. We can nicely distinguish the yellow, blue, and red cohorts. And that's where we are. So that's a typical metabolomics approach. Well, there's many problems with that. First of all, many people don't even realize which kinds of statistical tools they use. And there's a lot of overfitting, from missing gap imputations to actually supervised statistics and overfitting happening that would possibly lead to these types of groups. And secondly, if you actually work in human cohort statistical settings, you never get, or very rarely get, these three very distinct populations. They are much more overlapping because humans have huge within group variance. They're not like mice. They're not like plants. Humans come in all forms and shapes and sizes. So you don't get it this way. So the classic multivariate statistics is therefore not a really good way to do it. So what else can we do? If the multivariate statistics leaves us a bit in the dark, and the univariate statistics is also not a good way, what else can we do? Well, there is something. So you can use the raw values or p-values. You can combine them with regression models. You can combine them with Bayesian statistics. And I don't want to talk today about how to do this. And then you can perform so-called enrichment statistics. I will talk a little bit more about enrichment statistics, how it's done. The website is cambridge.us, but it's also embedded into MetaboAnalyst and also in the Metabolomics workbench embedded, different types of enrichment statistics. And the point is then you get very clear differences. And this is actually from the human cohort. And even with FDR corrections, because you don't do so many testings anymore, you don't do 1,000 tests, you do maybe 20, 25 tests of these different kinds of metabolic modules here, you can actually perform an FDR correction to that. All right, so the problem, of course, with that approach is here, in this case, you get chemical modules that are different between cases and cohorts, cases and controls, for example, such as unsaturated triacylglycerides or unsaturated phosphatidylcholine might be different. Now, these are chemicals, not pathways, not enzymes. And even if you relate them to pathways, that's not necessarily mechanisms. So you still have to do quite a bit of work after getting these types of statistical analyses. Now, let's assume we have gone further, and then people want to map things to pathways anyway. To which pathways? Many people would go right away to the Kyoto enzyme and gene network database, encyclopedia of genes and genomes, sorry, that has a long tradition in metabolism. And you in the consensus pathway DB, which is an overview, you get a lot more. You get wiki pathways, you get the small molecule pathway database, you get reactome. So you get a lot more than just KEGG. Not just biochemical pathways, but also, for example, drug-target interactions, protein interactions, metabolic reactions, and so on and so forth. So just give you one idea, like you can expand your point of view beyond KEGG to other pathways as well. And just talking a little bit about pathways, what is it anyway? Wikipedia is my favorite source of quick answers. Wikipedia says, a metabolic pathway is a linked series of chemical reactions occurring within a cell. Interesting. So how do these come about? The first pathway clearly was done by biochemical evidence, like the Krebs cycle. But today, pathways are generated from genomes or predicted from genomes in a semi-automatic way. And then human interpretations take these sets of chemical reactions and put a name to it, like glycolysis, TCA cycle. These are human interpretations because, the metabolites that we see, for example, citric acid, is not just in mitochondria, it's also very important in the cytoplasm. Pyruvic acid, the end product of glycolysis, is also found in the nucleus, where it's actually generating acetyl-CoA to acetylate genes. So you can't just find citric acid and talk about mitochondria. It's a lot more than that. And these pathways are intersecting just like street maps, like interstates, like streets in a city. So therefore, we have very many different ways to organize these and categorize these that you can see here. So, KEGG categorizes 537 pathway maps for 18,000 compounds. Brenda uses 110,000 compounds, a lot more, right? So obviously, there's much more information. Metapsych has about 4,000 fewer metabolites, but many more pathways, because in Metapsych, they define a pathway as uninterrupted sequence. So glycolysis, for example, has lots of interruptions. You can always go sideways, for example, to the pentose phosphate pathway or to other pathways. So that is a difference between definitions. And therefore, you can't just say, I map it to pathways, because you have to be specific. And apart from pathways, there are also, of course, other ways to map metabolomic data. David Wishart's lab in the Metabolomics Innovation Center in Canada have come up with a huge number of good databases, like the Human Metabolome database, 114,000 metabolites in there, FoodDB, 70,000, and DrugBank with 13,000. But there's even more compounds. So we have published the blood exposome database that has 65,000 unique structures that have been verified and published to be chemically detected in blood, many more than you would find in the Human Metabolome database, and all of these by literature, so from 900,000 literature references. And similarly, there's of course toxicology and environmental exposome research listed for 882,000 chemicals on the Comtox chemicals dashboard. So, there is a lot more than just pathways that we will also find in clinical metabolomic studies. All right. So going back to our question, what can we now do with our data sets? Now we understand that there's genomes and there's lifestyle and exposure, different actions of tissues and organs to our system, genetic interactions, protein-protein interactions, and all we got is the list of these metabolites and lipids and their abundances. And we really would like to know what is now driving what are the next steps we can do. So to all scientists here today listening who get overwhelmed, well, keep it simple. I try to tell it to all my students and to all my postdocs eventually you have to come up with something that people can understand. If you try to show all your data and all your figures, how do you even manage to convey a simple message, a story, from all these types of interactions? So you have to find your way through the labyrinth, where you find something that you can tell other people. And it could well be that your data set support more than one interpretation. That usually means you write more than one paper from the same data set. And that is, by the way, one of the reasons why it's not as easy as you think to download clinical metabolomic data sets for free with all the necessary metadata to interpret the data. So that's one of the reasons, because there is often more than one way to interpret the data. And for each paper, you need to find your way to interpret it. Okay, so how we do this, and this is now examples from our past studies. So we had a study on early detection of lung cancer. Lung cancer is bad for you. It's the most deadly of the cancers. The reason is because it is very late diagnosed and then leads to metastasis, with a huge number of deaths per cases. So if you look at the annual deaths for the by the cases in 2010, so that's like 3/4, right, roughly. So people usually who are diagnosed with lung cancer, they die. So what we did here, we looked retrospectively. We had 100 cases versus 200 controls as a discovery set, and 100 cases versus 200 controls validation. We had age, sex, BMI, smoking status, asbestos, the months status, like where they were exposed to asbestos, the months before the cancer diagnosis, the race, the cancer stage, histology, and so on. So we get a lot of metadata, additional information. Then we did metabolomics. That's really a long time ago. So at that time, we had 400 identified compounds and 2,000 unknowns. And working with Sam Manash at that time was at UW Seattle, we thought, well, what are we going to do now? Are we doing mapping it, multivariate statistics? What are we going to do? So imagine you would have such a full metabolomic data. What would you do? Because now that's a typical question. And so what we did is, because it was really a diagnostic question, we wanted to have early diagnosis. That's why we said, let's do the classic statistical approach, where just one compound is important to make decisions. So we used the HILIC data. The GC-TOF didn't do anything, so nothing of the classic primary metabolism was important. And at the very end of our HILIC run, there was an unknown, mass 287.245. We did not know what it is. We used the accurate mass and the MSMS to discern a sum formula. We then asked the database what it could be. And one of the answers was it could have been diacetylspermine. At that time, that standard was not available, so we synthesized the standard of diacetylspermine in our lab, and it was correct, right? Same retention time, same MSMS. This is diacetylspermine. Today you can buy it. You can even buy isotope labeled standards for this compound today, but at that time it wasn't. Why was it a good compound? It was actually higher in cancer versus control. That's the next thing. If you have a case versus control, it's much better to something that's not only very significant, even after FDR correction, very significant, but also higher in cancer versus controls. So that's one way, how we publish the metabolomic. Another study that we here did with Stanley Hazen from Cleveland Clinics. Here we looked at a much more complex disease, risk. So this is risk factors, including mechanisms. We used untargeted metabolomics again for cardiovascular health. Cardiovascular health means stroke, heart attacks, and similar diseases. So you see this, person. I always see myself here, like, walking a tightrope and try not to fall off, right? So here we had over 1,000 elderly subjects, age, sex, smoking status, but also HDL status, C-reactive protein, TMAO, baseline medication, hypertension, type 2 diabetes, background, and so on. At that time, we used lipidomics and HILIC. We had 600 identified and about 3,000 unknown compounds. And, we went from there. And again, what are you going to do? So, well, in these larger studies, you have to think about more than one paper. And sometimes, as an analytical chemist, you also want to be last author or first author, because as a professor, I also have to take care of my grad students and postdocs. So, for example, we use this data just for, method papers. And here we used only lipidomics papers, and we only used the identified compounds. And we published a paper, systematic error removal, using random forest to normalize large-scale untargeted data. By the way, that method works really fine, so please look at it. If you ever want to know how to do this, look at that paper. Another paper we published in bioRxiv was just using that to say, how big is the difference between male and females across human cohort studies? And that showed that it's really so big, the differences, that we now don't randomize anymore between men and women. We take them completely separate. We have separate sets for men and women because you cannot adjust for sex. It is not possible. So you have to run them differently. The lipidome and the metabolome is so different between men and women, even much more than body weight and so on. Okay. So, but that's of course not of interest to the clinicians. So here comes in another truth. Clinicians or the PIs may have a different way of looking at the world, and working with Stan Hazen, he's really interested in the interaction with the microbiome. That's really what's driving him. So you can call it a bias or a strategy or focus, depends on where you are. So the first paper he published is, okay, so trimethyl lysine is a strong predictor, but trimethyl lysine itself is not a source for TMAO, which was published by Stan Hazen before as being really important for cardiovascular health. All right, that's fine. But then he published, he used the data and continued with the data. He not just published another compound that was of huge importance, but he then looked into the mechanism. And last year, he took the data and published in Cell, so that phenylacetylglutamine not only is a big predictor for heart disease, but also how it works. So it's microbial produced and works through adrenergic receptors. So it binds to adrenergic receptors, and that's the mechanism how we can understand why in this case for this population, how certain metabolites act on cardiovascular risk. So that's huge efforts, if you read that paper, much more than just having metabolome data. All right, another example, breast cancer. Breast cancer, the 5-year survival is much higher than in lung cancer, because also it's much more, many more therapeutic options available, except if you do not have receptor status, HER2, estrogen, or progesterone, which are typical medications that can act on these receptors. So what if you lack these 3 receptors? So what are your therapeutic options then? So that's a very different clinical question than just saying breast cancer versus not breast cancer. It's actually the question about, triple negative breast cancers versus, positive breast cancers. Here again, this is a really old study that we did 15 years ago or so, where we had age therapy grading, tumor percent, stage, receptor status, and at that time we had 3 unidentified compounds only. So okay, so we published several piecemeal results, like how to just use the lipidomics. That was actually with Matteo Resic at that time. Then we focused on very specific compounds, like glycerol 3-phosphate and the acetyltransferase expression that leads to buildup of lipids, because lipids are really important in cancer status, because you have very active cell division, and that means that you have very active acetyltransferase for this invasiveness of cancers. Then we just looked at central metabolism, classic hallmark of metabolism these days, we say. At that time, it wasn't known or not appreciated at this time. And then we, just looked at the ER positive versus ER negative breast cancers only, and looked into how much glutamine and the glutaminolysis acted on this breast status. But this is still not what we all want. We just take one thing at a time and publish one paper at a time. So for many years, we thought, isn't there another way to utilize the whole suite of metabolomics data? And what we did then is, the thing where people would immediately go, what about multi-omics? Why don't you use multi-omics? Multi-omics should be better than just metabolomics, isn't it? And specifically for this study, where we had actually tumor tissues and not just body fluids. All right, so let's try that. So we had here 300 subjects. All of them had either positive cancer status or ER estrogen receptor status or negative. And when we look at that, and we do now proteomics and metabolomics, and we only use the proteins that associate with enzymes or code for enzymes, then you get an image like this, a map like that, where you, for example, have as little circles, you would have the metabolites, right, like alanine here, pyruvate, and the hexagons would be enzymes, like pyruvate carboxylase, for example, and other enzymes. And then you can see upregulation is red and downregulation with blue. And you can make maps like, okay, cholesterol is increased. And so 2 enzymes in cholesterol pathways are increased very heavily. So, which is in itself an interesting finding. And so can you just publish that? We did not publish this. This figure was not published. Why? Because it is not quite easy to distill a single new discovery from it. So we then looked into cholesterol metabolism, asked a lot of breast cancer researchers, looked into a lot of papers, and this looks like a bystander, not a driver of cancer. So bystander metabolism or bystander pathways are a consequence of an alteration elsewhere, but not driving the cancer or the progression of cancer itself. And of course, a lot of it is already known in cancer, like nucleotide metabolism is, of course, hugely different in cancers, also ER negative versus ER positive cancers. So a lot, you can't really mess with that because, these nucleotide metabolism is important for any cell division. So it's not that straightforward to come up with new approaches that haven't been tested before and not published in breast cancer research before. So what we then said is we now need to take it, multi-omics, seriously, and we added text mining to it. So we basically asked from all the detected proteins and all the detected metabolites, what genes would be associated with these proteins and metabolites pathways. And we added then gene expression omnibus, where you can download all the genes that have been published to be differently expressed in breast cancer, negative and positive. So we took all gene expressions, protein expressions, and metabolite expressions together, and we just simply asked, what are genes that have never been studied? So these genes here that you find here on the left-hand side have never been reported. And other genes like CD44 thousands of times, and P53, that's not even on here on that list, would have been like 30,000 times studied. So the idea is this multi-omics can give you novel potential targets that are differentially expressed in and implicated in on the gene level, on the protein expression level, and regulating metabolic pathways that we have found. So that in principle, these might be novel targets. So that's another way how we think multi-omics can be used for clinical studies. Another study is here on lung cancer. We had here adenocarcinoma versus non-malignant paired tissues. So these are tissues from the same patients because you cut out very generously. And so you can always compare patient by patient using the each patient as his or her own control. So here's just 39 subjects. We had smoking status, stage and sex, only primer metabolism by GCTAF. So we had 178 identified compounds, and we can map them onto MetaMap software that we generated, basically putting metabolites into context both on biochemistry and chemistry together. And then we see upregulations in red, downregulations in green. We can look at that. So the good point here is every node has a relationship, clearly with chemistry and clear display of the regulation of biochemistry. But if you would now repeat this with more than one metabolomic assay, you get 1,000 metabolites. Such clusters can become very quickly very unwieldy. Big network, clusters, hairballs, we call them. So these work fine if you have 300 to 500 nodes, but at some point then, if you get more than that, you have to kind of collapse it and to get a better overview. So that is, of course, one option. And another one, of course, we tested is called correlation. So you can look at correlation, say glucose and glucose 6-phosphate are correlated, and glucose 6-phosphate and fructose 6-phosphate are correlated, and so on. But when you do this, you can also ask which metabolite correlations cannot be interpreted or explained by other correlations. This is called partial correlation. We showed this, and it's a way to reorganize the data set in very simple terms, negative and positive correlations, and also the metabolite levels, higher versus lower in cases of controls, and you get this. I had the problem that I really had hard times to go from this image to something that is an interesting interpretation. Some people still do use it. Even my grad student recently told me that he wants to revisit this idea. And I always said, well, if my result is I have 4-hydroxybutyric acid to be different, and it is positively regulated to with 5,6-dihydrouracil, what do I do with this information? Where do I go from here? And this is where I find these partial correlation networks not so suitable. because it's not easy for me to go from these networks to a deeper biological insight. And that's also, of course, something we need to have, and not just, saying, well, that's the result. All right, so again, going back, well, if we would do now this on the protein and metabolite level, we can put these nicely into maps, and we can put the maps also with names on it, like nicotinamide or NAD biosynthesis, 1-carbon metabolism, and polyamine metabolism. These metabolic pathways are really important in cancer, including lung cancer. And we see, again, the proteins and the metabolites being differentially regulated. We see the importance, for example, of specific bridge proteins. Here is the methyltransferase, a nicotinamide methyltransferase that we published in another paper with another researcher subsequently, as one of the drivers, actually, of certain cell studies. So this is good. But the problem was here, there is no software giving you that clarity easily. So this is all handmade. This is just a person, in this case, my postdoc, Johannes Fiehn, at that time, just sitting down and making a nice graph in PowerPoint. So I thought, like, okay, well, that's funny, because you would hope that there is somewhere a software you can just submit your data and you click it and you get this out. Well, if it was that easy, I'd be happy, because this is after a lot of thinking. It's already interpretation that goes into this kind of graph. All right, let's go back to what else can we do. And as I said, we have the opportunity to combine p-values with context. And this is called set enrichment statistics. So imagine you have all metabolites in a database, and you can, put sets on top of the database. A set could be a pathway. Glycolysis is a set of metabolites in the KEGG database. Or you could say triacylglycerides. This is a chemical definition. So you can say, I define a set of triacylglycerides as being important. And then you can look at your measured metabolites, including their raw p-values, and ask the following question. If this is the number of metabolites that are significant in my blue set, how likely is that this is an over-enrichment, that I wouldn't expect that by chance? This is a very different question now than just the raw p-value. And the reason is classic statistics would now take every single individual compound and say and scrutinize it with false discovery rate FDR corrections, as I have said before. However, if we now see in a certain experiment we had so many metabolites in that set, this is not by chance. So while every single metabolite might only be having a 0.05 or even 0.1 raw p-value, altogether, it is unlikely they would behave in this way. And so then we can do statistical assessment. How likely is that? And this is leading us to set enrichment statistics. That's the idea of set enrichment statistics. Now, obviously, it is very critical to think about what are the compounds in my set. So if I use the pathway database, well, which one? KEGG or Metapsych or whatever, because that defines the blue circle of my set. Next question is, pathway databases are sparse. Lipids are not well covered in KEGG, for example. So, if I have a lipidomics data set, KEGG is pretty useless. And another one is, I have many compounds that may be represented in multiple pathways. Think about ATP, glutamate, NADH, and many, many others. So you would double count, double dip. That's not right. That's not useful. And for that reason, in my laboratory, we never use pathway set enrichment. Instead, we use chemical set enrichment. All identified compounds are included because they have a unique structure. Structures can be automatically classified by using substructures. Easy. Our software also allows you to label things manually if you are smart. And you say, well, these compounds, apart from I know the chemical structure, I also know where they come from. For example, from food, or these are pesticides, or these are drugs. You know, you can put them into label like exposome or microbial. And you can say, well, I can have my overall statistics, the umbrella, the blue stuff, defined by the experimental data. So that you would ask, of the set of all the triacylglycerides, how many were significantly different, rather than asking of all triacylglycerides in the universe, right? Because that is a much bigger blue ball. However, of course, when you get the answer, you get an answer on triacylglycerides and not a direct link to genes or enzymes. So this is an example on the last study I want to present today. ME/CFS patients. This is chronic fatigue or exhaustion. This is very, very similar to long COVID haulers, so therefore huge interest. We have not published this particular image yet. We have presented it at conferences, but not published, and we're working on the publication with Ian Lipkin at Columbia University. And what we now see is, again, huge differences, decreases in blue, for example, for phospholipid ethers and plasmalogens, which are also ether lipids, but also unsaturated ceramides, phosphorylcholines, lysphosphorylcholines, lots of unsaturated fatty acyl lipids. Not so many primary, some dicarboxylic acids, but not many. Huge increases in unsaturated long-chain triacylglycerides, but decreases in saturated triacylglycerides. So with these information, now you can do interpretations. All right, actually, it wasn't my last study. I have another one. Because often in clinical studies, you look for phenotypes and not just for positive, negative case control. And association, so you look at association of a metabolite to a phenotype. And here just take heart disease as a function of biking to work and smoking, just from the internet. So give you an idea like, okay, we look at this trend rather than plus minus. We use this idea for the Alzheimer's disease neuroimaging initiative with Rima Kaduradog, where we have cognitive normal, mild cognitive impairment, and people with Alzheimer's disease. And we then used the metabolomic data to associate with imaging data, with clinical data, including the cognitive assessments, and of course, markers for Alzheimer's disease. So these markers for Alzheimer's disease is, for example, the peptide clearance, the accumulating tau tangles, brain atrophy, cognitive decline subscales. And so we now just used here the lipidomics data set. We had linear regressions. So now we associate every single lipid with one of those phenotypes, including, for example, sex, but also body mass index, age, right? ApoE4 status, which is a genetic mutation known to be associated with Alzheimer's. And these are all FDR adjusted. So that's another way how to do statistics. And you look at that and you say, well, I see a lot of red and blue. Well, first of all, you see typical stuff that you would expect. BMI levels, body mass index, is clearly associated with triacylglycerides. Ta-da! Very, very interesting. But we also see, for example, other things like, well, first of all, differences between sexes. Well, I said that before, but also other things, scattered acylcarnitine, ceramides, and so on with tau tangles, positively associated with tau tangles, both then stratified by Alzheimer's or cognitive normals, and a mild cognitive impaired. So these are the color codes here. So how do we get better than that? So the first thing we then used is again set enrichment statistics. So that simplifies this big image into a clearer image, where we now have the acylcarnitines and the glucoceramide and the triglycerides, associated with these phenotypes. And interestingly, now you see red and blue being in different directions associated with the diagnosis of Alzheimer's himself, but also, of course, with specific compounds associated with mild cognitive impairment, but not with complete Alzheimer's, and so on. So we were still not satisfied, with these, both up and down regulated. How do we interpret that? So we went further and adjusted this Cambridge association with phenotypes even further. And we came up with this. So instead of looking just at the head group of lipids like phosphatidylcholines or cholesterol esters or ethanolamines, we were asking about the acyl groups, specifically the polyunsaturated fatty acids. And then we saw very, very clear differences. For example, EPA-related or DHA-related polyunsaturated fatty acids were hugely associated with fish oil intake. Yes, because many diseased people use these fish oil capsules because they think it might help. However, we also see that although these people took the fish oil supplements, were not incorporated into these complex lipids. So they were negatively associated with diagnosis. So it's really, a conclusion here is the supplement diets didn't help. And in fact, clinical interventions didn't help either with cognitive impairment because the enzymes don't work. The acyltransferases don't work that lead to the incorporation of long-chain fatty acids into the brain. And then we see also much clearer associations with, for example, spare-AD and tau tangles and so on. So this is what I would like to give you today. You can also then, of course, do individual clusters of lipids with the tau tangles, positive associations, the same cluster negatively associated with some other phenotypes. So that is another way how we can interpret human cohort studies beyond just pathway mapping. A segue to Lloyd Sumner's talk, don't dismiss unknowns. This is from my past. Plant studies. In plants, we have a much more intersection of identified compounds, and unknowns are much more species specific. And I'm sure Lloyd Sumner will tell you a lot more about compound identification in plants. And so overall, conclusions: never ignore the underlying biological context, discuss a lot, take steps to understand the broad and interconnected role metabolites play in the system, define the limitations, and do not overinterpret your data. And so multi-omics is usually not the answer, except for maybe cancer biopsies, right? So usually it's really difficult to do and not helpful necessarily, or definitely not easy to do in biofluids. Pathways have very varying definitions. Metabolites also give you information on foods, drugs, microbial, environmental. So lots of other ways how you can interpret data. Online databases give you lots of insights. And for unknowns, you can do a lot more. Some of that you have learned from Arpana Vania. So I'd like to thank all my team members all my collaborators, some of them I have had before, and I'm happy to answer questions.
Great, thank you, Dr. Fiehn. We do have some questions in the chat, and I'm sure some more will appear. I could actually start with a question from Siju.
Okay, humans are
So,
unlike plants. Yeah, this one?
yeah, go ahead with that.
Okay, so humans are unlike plants, which eat carbon dioxide and water. We eat everything. You look at food markers. Not you want to find metabolites made by, not by food. How many metabolites do human cells synthesize? 114,000 metabolites in the database may include many from food. Humans are not human. Humans are an environment, ecosystem. So we do not make vitamin C because we can eat it. So to distinguish the food metabolome from the human metabolome does not make sense at all. So to restrict things in plasma to the human genes doesn't make sense because we have microbes. We cannot live without them. So therefore, the human metabolome is not defined by the capacity of enzymes, full stop. I would like to say that very clearly. What is the standard to judge whether the metabolite can be seen as differential metabolites? Which is more convincing, fold change or p-value? Very good question. Every good statistician will always tell you they only give you a number. And in my book, it is the fold change that I like the best, because significance just means worthy a second look. And if you have something that is a 5% change or 2% change, and it's significant, I wonder if that can be meaningful. So fold change, twofold, is often meaningful, right? So, however, in clinical studies, of course, you may look at the population, and then maybe a 5% or 10% change may be important. So it depends a little bit. Okay, so I'm wondering about the reproducibility of metabolomic results across different cohort studies. Good point. Excellent point. Many people wonder about it. There are efforts on the way to improve the status of untargeted metabolomics by making untargeted metabolomics more standardized, using kits of internal standards. So in my lab and in multiple other labs, we have also using multiple types of controls, pooled quality controls, commercial quality controls, and NIST plasma or other NIST community core controls, in addition to internal standards. For example, in lipidomics, we use the enriched set from Avanti that now has, I think we use 96 internal standards. So there is progress, but it's certainly not easy. Okay. Can you describe in more details the underlying databases and standardizations that are necessary to enable large cohort and multiple study analysis? It's a lot of work. That's why you need postdocs and time. So often enough, you end, the funding for a project ends, and somehow the agency expects you to have published all that, but the last data come in year 4, sometimes year 5. So obviously it is not easy to do that. Standardization, the interaction between the databases is not easy. The discussions take time too. So very often we publish papers years after a cohort study is completed. We've done it on the breast cancer studies. We have done it with Stan Hazen in cardiovascular risk, very, very often. It is not nice to do it because then your postdocs have left and you scramble like somebody else who like to talk about it because that person who originally on the project is gone. And that's why we have the principal investigators who try to keep their head above water and try to keep an overview. But yeah, it's different. I cannot give more details here, really. That would be a different, I guess, enterprise. So with that, that's all.
Yeah, I actually have one question for you, Dr. Fiehn.
Okay, sure.
So you described set enrichment, and to me that sounds very similar to using gene ontology.
Yes, it's borrowed from gene set enrichment.
Yeah.
So there they used gene ontologies as a set definition.
Yeah.
And we use also the same type of statistics called Kolmogorov-Smirnov statistics.
Mm-hmm. So for me, using Go analysis, it always seems like I get the most clues from the functional studies, which to me seems most similar to pathways in metabolomics. But you're saying that pathways are not the way to go. Do you think that's a fundamental difference between the metabolome and the genome? Or do you just think it just reflects our state of knowledge that we know so little about the full metabolome?
Well, yes. First of all, when you do genome analysis, you look at one organism, say a cell or a tumor or so, or an insect. There's biliosis, like, what about insects, right? So and you have a defined set of genes. In metabolites, you do not have a defined set of metabolites because humans eat. It's in plants much easier because Lloyd Sumner will talk about plants in a moment, because there, the plants don't eat. In general, some plants do eat, but that's a different thing. But most plants don't eat. So that's a first huge difference that I cannot stress enough. And the definition of a function of a metabolite is something we don't have yet. So for some metabolites, we understand what they do, like glucose is used for oxidation and making energy. But for many compounds, we don't exactly know what they do, other than they are involved in signaling, which is like hand waving, right? Mm-hmm. Like sphingomyelins and ceramides, forming lipid rafts. So that's their function. The function is not sphingomyelinase that converts ceramides to sphingomyelins. That's not the function, right? It's an enzyme in between. The function is forming lipid rafts. Or, for many compounds, we just don't know yet. And that's one of the reasons why we use phenotype assessments rather than just relying on pathways, or let alone genes and enzymes.
Yeah. So I have another question, which is kind of speculative, but I'll ask it anyway. So with regard to proteomics, I mean, now we have DeepMind and development of, structural prediction for proteins that we never would have imagined like 5 years ago. Do you think that's going to become analogous to your chemical structure sets, and that those kinds of analyses could be done on the proteome?
On a metabolome. So predicting, for example, first of all, the mass spectra. Predicting NMR spectra works fairly well, actually, notwithstanding what Voigt might say. But, predicting mass spectra is not easy at all. So in my book, machine learning has failed spectacularly. So some things can be predicted nicely by rules, like lipids. Lipid spectra can be very nicely predicted. Natural product spectra cannot be nicely predicted. So we use quantum chemistry, and we have published a couple of papers on quantum chemistry. And last week I told my grad student, wouldn't it be fun to predict 100,000 spectra by quantum chemistry? So far we have done 100 and 800. So, I said, then it's just more clusters to do 100,000, more computational time.
More computational.
Right? I hope that we'll publish that in a year or two. But definitely, it's a huge thing to be able to predict not only structures, but maybe also functions.
Yeah.
Maybe docking. I love to see the toxicologists better predicting who loves whom, like which metabolites loves which receptor. Oh my God, it would open so many doors, right? The the toxicologists would rather say, like, docking in terms of a ligand interaction on the catalytic site, not just somewhere on a protein. These are where we hope that other fields will help us in interpretation of data sets.
Yeah, absolutely.